Hi, everyone. My name is Lisa Galea, and I'm going to actually start the program because I know you don't want to listen to me. You want to listen to our amazing speakers. But before I begin, I want to do a lang acknowledgement. I want to acknowledge and recognize that we live, work, and play and participate in a community on the unceded and traditional ceded territories of the 203 First Nations. This is in Canada, of course, along with the 38 Métis chartered communities, each of which possess their own unique traditions and history here on this land that we now refer to as British Columbia. But I recognize that many of you are calling from outside of British Columbia and outside of Canada, so I encourage you to learn about the Indigenous peoples on lands of which you reside. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action, the United Nations Declaration on the Rights of Indigenous Peoples, and the BC Declaration on the Rights of Indigenous Peoples Act. In all of our work, we're committed to ensuring the Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, myself, I'm joining you from Vancouver, which is part of the unceded homelands of the Coast Salish peoples and the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. And we are part of the Women's Health Research Cluster. We're bringing this workshop today from a lot of work from one of the working groups known as the SGBA working group, which stands for Sex Gender Based Analysis. We have a number of cluster members. It's free to join, so please do become a member. And we present a number of events along, such as this workshop that we have here today. We also have a hormonal Women's Brain Health Conference coming up in November. It'll be virtual and it will be on hormonal contraceptives. So please look out for some announcements about that. As well as our seminar series and trainee presentations that will be coming up in a little while, I think just maybe even the part of this week. So take a look at our website for information about that. And of course, we can't do this work without sponsors, so please feel free to donate if you're so enabled. So we have three amazing speakers today to talk to you about how to SGBA. And we first have Jennifer Williams, who is our moderator for this session. So Jennifer, take it away.
Hi folks, my name is Jennifer. I'm from McMaster University. I'm thrilled to be first sharing our first speaker, Dr. Annalise Beery from UC Berkeley, who studies the neurobiology of social behavior, impacts of experience on development, and the importance of using diverse organisms, both sexes as well as multiple species in biological research. In the latter area, she's contributed to multiple efforts to promote inclusion of females as research subjects, as well as commentaries on the importance of comparative research. So I'll ask Annalise Beery to share your screen now, and we welcome you for your talk.
Looks great, Annalise.
Great. The unmute button, of course, shifted and I had to find it. Can you all hear me okay?
Yes.
Okay, great. Wonderful. Okay, so since we're all in Canada virtually, I thought it only fitting to borrow this fantastic backdrop from the Canadian Institute for Health Research. And I'm going to talk to you a little bit about a then and now in terms of sex inclusion in the biological sciences, and then talk about issues relating to how do we analyze our results by sex, which is really what we're all here for today. All right, so a long time ago in a galaxy far, far away, also known as 2009, I conducted a survey with my former graduate advisor, Irv Zucker, of sex bias in subject use across many different disciplines. And this is a reprint of one of the graphs from that paper focused, in this case, in non-human mammals. We also looked in humans. We also looked across the last century of sex use. But what you can see here is if you look at the bars, first of all, gray is not even declaring what sex is used in the study. There are an awful lot of studies that are not reporting the sex of the subjects. And then when sex of the subjects is being reported, these are, remember, mammal studies, more studies tend to use in most of these disciplines male-only studies than female-only studies, and often then studies of both males and females. So you can cruise this bar along and look at other fields. My own home field of neuroscience was one of the worst offenders, with 5.5 male-only studies to every female-only study and a minority of studies using both sexes. And you find that some other fields, for example, reproduction, probably intuitively tend to capture the sex of the participants better. You don't tend to write reproductive articles without mentioning what sex you're doing them on. And then studies conducted in the field of animal behavior tend to mostly focus on both sexes because that's what's out and around you. So I just wanted to highlight some of the differences. We found male bias in 8 out of 10 fields surveyed, and many, many other documentation of this have occurred. So this is found repeatedly in specific fields, and it's also found across fields with various different approaches over time. All right, so that was a survey of papers written in 2009 and even further back. Have things changed? Well, in the US in 2014, the NIH announced plans that sex as a biological variable would need to be an important consideration in future research. This was implemented in 2016, and I have a little logo here. There have been many other implementations across the world. And how does that impact our research? Well, what you can see across disciplines from 2009 to 10 years later in 2019 is that we do in fact have a significant increase in studies including both males and females. So that's good news. If we go over and look at behavioral physiology here, or sorry, that's neuroscience, you see also that same sort of significant increase. So most of these fields and all of the worst offenders had a significant increase in inclusion of females. So I'm going to put a little party hat on that one. However, before you get too excited, I also have a wet blanket, and that is that of the studies that were including both sexes, there was absolutely no improvement except in pharmacology, which had the least increase in sex-inclusive studies, of how those results were analyzed and whether or not sex was included as a variable in analysis. So I would say that sex inclusion, we can have that celebration, but in terms of analysis by sex, we have a lot of work to do. And I'm going to further highlight this recent study, the Rytz et al. Study that shows that yes, they also found that same increase in inclusion of females. But if you go further and you say, how were those females included? Did you have a balanced design? And were they included in all the parts of the study? Also, not getting a big improvement here. So we have a lot of room for growth in terms of how sex is analyzed in our studies. So that's just the backdrop and the foundation for today, where we want to really focus on how do we improve our analysis of research that's inclusive of males and females. Okay, so the first conclusion is not rocket science. It's analyze your results by sex, except that it turns out this can actually be harder to do than you might think. So our first question today is, how do we do these kinds of analyses? Well, when we're doing analysis by sex, at the big picture level, we have a few challenges to think about. One is, what if we make too big a deal out of sex differences? This is a really lovely paper by Donna Maney, focusing on sort of how do we talk about sex differences and how do we articulate them in a way that conveys how big they are and how not big they are. What if we fail to detect important sex differences? Many of these sex differences have really important, potentially clinical applications. And if we miss them, that's a big problem. And then we can talk about, sort of what if we are not careful with our language and our causality and our discussion of all of this, and we mistake sex and gender or correlates of sex and gender. So those are just sort of in the background, some potential things to keep in mind. Now, there are many, many guides out there to how to do this kind of analysis. I'm showing you a figure from one that I participated in construction of, but certainly there are many different guides to help you do this. But one way to think about this is the main thing you want to do is you want to look for your sex differences, right? That's the first sort of foundation here. And we want to describe them so that we get as much information as possible about those sex differences. So let's say you're doing a study and you find sex differences. They might be over on this far left side where they're so big and so obvious and you have such different effects in males and females that you don't even necessarily want to quantitatively compare your males and females. Maybe the males and females have different reproductive organs. Maybe they have an entirely different pattern, right? In that case, you're going to be analyzing your results separately by sex. So that's over on the left side. A lot of the time, we're going to be in the middle here, where we have some sort of quantitative sex difference, and we're going to want to make sure that we include sex as a variable of interest in our analysis. And the reason I say variable of interest, not just variable, is that lots of human studies in particular will include sex as a random variable and kind of try to factor it out. of a study and say, once we ignore the effect of sex, here's what we get for everybody else, right? But here we want to actually include sex as a variable of interest and describe the effect of sex. And we want to describe those differences and how big they are. So here I'm taking a figure from Donna Maney's paper, which is a really nice visualization of sort of the difference between we have significant sex differences and we have big sex differences, right? You describe the effect size and visualize that difference between males and females. You can really see how big is that effect and how significant it is. And those are going to be different. Maybe you had a teeny tiny study and you still found a big sex difference. That means a different thing than you had a huge study and you found a very subtle sex difference. Those are both important to note, and they would be noted differently by including effect size in our analysis. Okay, and then finally, over on the far right of this graph, we do in fact account for the fact that many people like to pool their male and female data. As much as I might like to think that that's not usually the right approach, sometimes, sex differences turn out to really not be an important issue. And then as a secondary thing, once you've done your sex-specific analysis, you can go ahead and conduct analyses with sexes pooled. So this is our little flow chart. Feel free to pop in with different opinions later in the discussion. All right. So I also want to highlight that picture sort of encapsulates what do you do after you have your data. And think about research. We need to think about this before we design our studies ahead of time. So as we design for these studies, we want to kind of think, is this just a study that includes males and females? Or is this a study that's actually designed to be sensitive to sex differences? And that's something that's going to come up in our next talk. We have both Ariane and I have done some work looking at sample sizes and how you need to plan for different sample sizes for different kinds of goals. And this, I've stolen this image from a paper that came out of Sabra Klein's lab very, very recently. It just sort of nicely encapsulates that at all these different levels of analysis, whether you're doing cell culture on up to population studies, you have a choice to make. Are you simply including males and females and considering sex as a biological variable? Or are you powering your study to look for sex differences, and do you have hypotheses about sex differences? Are you going to explore the mechanisms that underlie these sex differences? And although at this cartoon level, that's sort of stratified into two options, really your study could be anywhere in between. And I often find that my studies are somewhere in between those two goals. So the request was that we include examples from our own work. And so I'm going to just show you a sample of going through these decisions in a study in our lab. It's not easy, and here's why. So here's a recent study that came out, a paper in our lab came out this fall, and we were studying social motivation in vole species that form social bonds. So part of what made this challenging is that we had a lot of different comparisons we wanted to make. We wanted to know, the main goal of the study was to find out, does social motivation vary with familiarity? Does it vary across type of relationship? So these particular voles, the prairie voles in particular, form partnerships with mates. They also form these companion relationships with what we call peers. And we were very interested in knowing if those were different from each other. We wanted to know if these peer relationships varied across species with different social organizations. So we sort of had this framework of all these comparisons we wanted to make. And then on top of that, we wanted to overlay, does it vary by sex? And familiarity isn't captured in this picture, but that was the other sort of variable we were interested in. Okay, so what do we know about potential for sex difference in this kind of paradigm? Well, nobody has ever tested the effects of familiarity on social motivation before. And that's often the case as we do research, right? Nobody's done the particular thing that we want to do. So we don't know if there's a sex difference. What do we know about other sex differences in voles? Well, sex differences in the partner preferences they form, where they like to be with a vole that they know over a vole that they don't know, they're very minimal, and many studies don't find them at all. But there are some subtle hints that there are factors underlying the partner preference in this species that are different. So they get impacted differently by different manipulations. They have differences in the underlying mechanistic pathways, at least to some extent. So we've got indications of maybe some latent sex differences in this particular species. Okay, and then we have our study design. We're going to train animals to see if they'll work to access other animals. So we train them with the food reward first to make sure they understand the setup, that they're all pressers. Then we put them in the social setup where they can press a lever go on over to the other vole, and then they like to sit on top of the other vole for a minute, and then we shoo them back over, and then they can press again for more social access. So, and then we have this extensive timeline where each animal undergoes at least 60 active days of testing because of all of the training involved in this, because of all the different paradigms that we did, because we did all of these extinction controls afterwards. So it's a long-term hefty study, which is part of the reason I also chose this one, is we really wanted to shrink our sample sizes down as small as possible, but we really wanted to enable discovery of the things we were interested in. So in the end, we ended up with 5 different groups. And in meadow voles, we only study females because they're the sex that shows a difference in sociality across day length. So in not all cases did we study males, but in the prairie voles, we studied males and females for each of the conditions, and we had same sex and opposite sex conditions. And then normally in a study of this type, where you're doing this very repeated testing, the sample sizes are quite small. So most operant social studies are 4 to 8 in terms of sample size. We went with 8 because we were nervous that we wouldn't find something if we went down as low as 4. And we went with 8 in the males and 8 in the females for each of those groups. So this is a case where for us, we really wanted to be able to analyze our results separately by sex in case there was a sex difference. And so we had full size, but not extra size animal groups. And then this is just an example of the reporting of how do you sort of account for what do you get in the end results. And lo and behold, males and females showed qualitatively different response patterns in the social chambers, as well as significant interactions between sex and the variables of interest in a model screening for sex differences. So we looked at sex by stimulus type and sex by stimulus for familiarity. So further responses were further analyzed separately by sex. So that's sort of the boat that we ended up in this particular study. Okay, my timer has disappeared off my screen. How am I doing on time?
You're minus 3 minutes.
Minus 3 minutes. In that case, I'm not going to talk about this issue at all. I'm going to leave on my conclusion slide, which is what about cycle phase? Just like everything else we're going to talk about, whether or not you need to include females at all cycle phases depends on what you're trying to do with your study. And hopefully we can talk about this issue later in the Q&A. Sorry, I went over.
No, perfect. This is what always happens, right? We're just trying to give people a flavor. Jennifer, do you want to introduce our next?
I do. So thanks you so much, Dr. Beery, for that presentation. I'm looking forward in our discussion after to talk a little bit more about phases, because that's what I study. All right, so our next presenter is Dr. Ariane Albert, who's a senior biostatistician at the Women's Health Research Institute and has been working with WHRI for the last, since 2011. Ariane has a PhD in zoology from UBC, about 15 or so years of experience with experimental design and analysis encompassing many areas within women's health research. And her specific interests, as we'll learn about today, lie in elucidating best statistical techniques and study designs to answer questions in complex and observational data. So take it away, Dr. Albert.
Great, thanks, Jennifer. I think I'm going to be pretty brief, so hopefully we'll have lots of time for questions at the end. Thanks for the introduction to what I was going to be talking about, Annalise. So I'm going to go into a little bit more detail about the sample size and power issues that we may come up with when we want to include both sexes in a study. And I know this is a sex and gender-based analysis workshop, but for this particular talk, I'm going to just be focusing on sex as the biological variable in analysis. We can talk about gender issues later. Okay. So the question that comes up, and I think it came up in the questions for this workshop that people submitted, was, do you need to double your sample size when you include both sexes? So we just saw an example where Annalise was designing an experiment in her lab, and they did double the sample size for including both sexes, but do you always need to do so? And the answer I'm going to give you is the answer that statisticians everywhere like to give everybody, and that is that it depends. So it depends on your study question, it depends on the particular animals or humans that you're working in, it depends. So what does it depend on? So let's do a little thought experiment here. Let's pretend that we are designing an experiment to look at the effect of some new treatment in chickens. Okay, I just chose chickens because we could see a difference between males and females easily in a picture here. So we've got an icon of a rooster, that's the male, and a hen, that's the female. So let's imagine we're testing some new treatment for, I don't know, getting rid of worms, getting rid of parasites. Okay, so what do we need to think about if we wanted to include both sexes in our experiment? And the first thing we need to think about is, what do we expect the effect of sex might be? Now, Annalise has just shown us an example where they didn't know ahead of time what they might expect the effects of sex could be, but we can come up with some potential examples of what we might expect that to be. And before we dive into those details, I'm just going to introduce a little bit of terminology here. So Annalise was talking about interaction effect. And in the simplest possible case, you can imagine you've got an experiment with two variables. So one is your treatment or your group differences or whatever it is you're looking at may not be an experiment. The second one is sex. And let's imagine we just have two levels in this case for sex, male and female. The interaction effect is a specific statistical case where you expect the size or the direction of the treatment effect or the difference between your groups to be different between the sexes. I'll go into a little bit more detail to explain exactly what that means, because that's a lot of the words different in the same sentence. Contrast that to a main effect where you have still the two variables that are of interest, treatment and sex, but now you're only interested, is there a difference in the sexes in your outcome variable, whatever that is. In our case with the hypothetical chickens, it would be parasite load. Or are you interested in differences between the treatment groups, control and treatment, in their outcome, parasite load? But in this case, you're not actually testing for a difference between the sexes in that treatment effect. Okay, so there could be differences between the sexes, there could be differences between your treatment groups, but you're not investigating differences between the sexes in their treatment effect. Okay, so what does that look like? Graphically, it's a little bit easier to explain if you have some pictures. So let's say we gave half of our chickens some anti-worm medications, and we saw that in both males and females, they had the same average level of reduction in parasite load. Okay, so this is the same effect in males and females. The size of those arrows is the same direction. We would call this a main effect. And I've just put in a little figure over here on the right so you can see what that might look like. Use the control group, males and females are very similar. Treatment group, males and females are very similar. The size of the difference between control and treatment is the same in both sexes, okay? In this case, you wouldn't necessarily need to increase your sample size, but you would want to include equal numbers of males and females. And you may also want to analyze the main effect just of sex in case there is on average a difference between males and females, let's say in their baseline parasite load or something, for example. Whether you want to do that, again, depends on your particular study design in question. So let's go to the more extreme example, which would be a huge interaction effect, which would be something where Annalise was talking about. You could have a completely opposite effect in your males and females. So let's say, contrary to all of our expectations, when we give this anti-worm medication to our chickens, female chickens actually end up with an increased parasite load and male chickens end up with a decreased parasite load. That would be weird, but let's just roll with it. What does this look like in terms of like a graphical representation of your results? Well, your control group, you would expect, no, there's nothing happening. They have similar parasite loads, but in this case now females in the treatment group have higher parasite loads than the controls and males have lower parasite. In this case, you actually don't necessarily need to increase your sample size. Depending again on what your question is, you would want to include equal numbers of males and females. You would definitely want to test for an interaction. And you would probably want to analyze this by the sex separately, which is what Annalise was talking about earlier as well. You would probably also want to include enough of each sex to estimate their treatment effects within their sex group, whatever the size of that treatment effect would be. So you'd almost essentially be designing two separate experiments, one for each sex, to make sure that you were powered to detect the effect in each one. What would happen if you ignored sex? Let's say you just analyzed, you just forgot that sex was a thing and you just analyzed for your treatment effect. Well, in this case, you would actually end up with a treatment effect that was halfway between that for males and females, which would suggest to you that your treatment was completely ineffective, right? Your average for that treatment group would be that it would be halfway between the males and females. And if you only looked at one sex, let's say you only tested this treatment in males, you would think it was fantastic and you could go out and give it to all of your chickens and you'd end up with half of your chickens having an increased parasite load. That would be terrible. So in this case, it's actually really important that you think about sex and include sex in your analysis. Now, there's a lot more potentially subtle differences that you could have between the sexes. So what if there's just a smaller effect in females compared to males? So the medication is good for clearing parasites, but not as good in females. This is an interaction effect again, but it's not as profound as the one I just showed you. So in this case, we could see that males have there's a good effect in males, less in females. In this case, you would absolutely need to increase your sample size, possibly double, possibly triple, possibly even more than that, depending on what the difference was between the sexes and their treatment effect. And again, if you ignored sex and you just went for the average treatment effect here, you would have lower power than you would have if you had only done a single sex study. And again, using only one sex would give you misleading results for the other one. So regardless of which sex you chose, in this case, if you studied the drug only in females, you might think that it wasn't going to be effective at all. Whereas in fact, if you used it in males, it would be amazing. So these are important considerations. And just a quick, brief look over some design implications before we can actually have some interesting examples from Liz's work in a minute. So what do you have to do? How do you calculate these sample sizes? What do you actually have to do? So you essentially need to decide on what your minimum treatment effect or minimum difference between groups, whatever your experimental design is, what is that minimum treatment effect that you would want to be able to detect? Okay, so let's say, again, we're looking at parasites. If you reduce the parasite load in half, is that a big enough effect of that treatment to make it worth using that treatment in the real world? What's the clinically relevant effect size? If you're doing behavioral experiments, obviously that's going to be different. I am a bit more focused on the health intervention side here, but essentially, what is the difference between your two groups that you would consider to be relevant? You need to power at least for that. And then on the same side of that coin really is how large a difference between the sexes would be relevant, right? Because you could have a difference in treatment effect between sexes, but if it's really small, let's say females, they're only a quarter different from males, right? So they have 3/4 of the effect size. Is that clinically relevant? If it isn't, then that's an important thing to know. So how big of a difference between the sexes would you consider to be relevant? And then you would want to power for that difference. And you can determine your sample size based on these two values or these two competing interests, essentially. So if we had a single sex study, let's just say we had again, let's set our parasite load, 5 worms in our control group. Oh no, that doesn't work because we're going up. Something else. Anyway, so our control group is 5, our mean in our treatment group is 5.8, standard deviation is 1 in both, super easy. A treatment effect in this case is 0.8, or what we would consider to be a fairly large treatment effect in a medical study. To get 80% power to detect that difference, we would need 26 animals per group, or a total of 52 animals, which is a lot, but for something that had this kind of treatment effect, it's not unreasonable to expect you would need that number of individuals. So what would happen if we wanted to include the second sex, and we wanted to at least be able to see if they had half the treatment effect? Okay, so the difference between males and females in this case is that the mean in the second group is half of that. The treatment mean has half the difference from the control mean, and one sex is the other. So the treatment effect in this sex is 0.8, and in this sex would be 0.4. If we kept our same sample size and we wanted to test for that interaction, we wanted to see that half difference, that there was only half the effect in one of the sexes, our power would only be 12% for that effect. It would be 60% for a treatment effect ignoring sex. Of course, your estimate for that treatment effect wouldn't necessarily be accurate, right? It's going to be halfway between the one for each sex. So what happens if we double our sample size here? If we did 52 per group, our power is still terrible for the interaction term. It's great for the treatment effect. So, if you were just interested in estimating the average treatment effect across both sexes, you would be fine in this case by doubling your sample size. But what if half the treatment effect is actually something you were interested in finding out about? I kind of think that would be potentially important if you were designing a new intervention. So again, for this, just to recap quickly, 0.8 a treatment effect in males, 0.4 in females. So it's half the effect in females. Even with 10 times your sample size that you started with, the power is only 60% for that interaction term. In this specific example, if your treatment effects are larger, okay, so let's say it's massive, it's 2 in males and 1 in females, it's still half in females, right? But now you have a much better chance of detecting that interaction term with a sample size of 60 per group. So not insanely much larger than it was for the initial one. So really what I think the take-home message here is that you really need to have good estimates for what size of difference you would be willing to miss. Okay, so what you would think would not be necessarily relevant between the two sexes. And that's what you need to kind of design your study around. And we can talk about the sort of nuances and implications of all of those soon, right? So let's move on to the next speaker.
Thank you so much, Dr. Albert, for that presentation. Really excited to get down into the statistics and modeling in our discussion. So I'll ask Dr. Elizabeth Rytz to share your screen. Perfect. So Liz is an assistant professor in the Department of Cellular and Physiological Sciences at UBC, and also holds a CIHR Sex and Gender Science Chair in Genetics. The overarching goal, as we'll learn about Dr. Rytz's research, is to identify specific genes and pathways that contribute to sex differences in metabolism, and how to understand abnormal function or regulation of these genes in the pathways that influence the risk of developing various metabolic disorders and diseases like obesity, diabetes, and cardiovascular disease. So take it away, Liz.
Sorry, everyone, my voice is it's a little bit off today, but it's really a pleasure to be here to listen to other folks and then to share a little bit of what we've done in my lab. So today I'll just be kind of like talking about more specific examples that have come out of my lab. And, you know, we're privileged in my lab because we do a lot of work on fruit flies, and we've just started to work on mice. And so these issues of statistical power in fruit flies have allowed us to find a lot of really cool stuff because we use hundreds of animals. And so we've been able to really detect stuff. And what I would say, if you remember nothing else from what I discussed today, it's that really the differences are quite stark between the sexes. And I would argue that, you know, even if you do detect at the level you're looking at, you know, not a big difference between the sexes, it's still possible that there's something different between males and females in terms of the underlying mechanisms. And I'll show you one example of that. So my work focuses on sex differences in metabolism and metabolic diseases. And over the next couple of slides, I'm just going to show you some data that we've had and the types of data that we get and the overall take-home message. I won't be talking about statistics or whatever. I'll just be talking to you about different ways maybe to think about the data that you might see in your lab or in your experiments. So first of all, if I talk about sex differences, people always say, well, like, how can I be really sure if I'm seeing a sex difference? And my answer would just be that it's any non-identical response to any genetic manipulation, pharmacological manipulation, or environmental manipulation. So, you know, things like diet, temperature, you know, knocking out a gene in this tissue and that tissue in the whole body, whatever. So any non-identical response is what I'm talking about. And in my lab, we've seen everything. So the most, you know, we kind of divide those into 4 classes. So for example, in class 1 here, you'd get a very sex-specific response, right? One sex doesn't care. Whatever you're doing, they just don't care. And the other sex, big response, small response, but they have a response. In class 2, this is really common. We see this a lot. So one sex cares a little bit, maybe. There's a small difference in phenotype in the purple bar here, but there's quite a big response in the orange bar, whatever that other sex is. So we see that all the time. So very sex-biased response. Class 3, I have to be honest, there's a couple of genes that we've seen this for, but we don't see it too often. And statistically, it is, you know, people have done studies, it's less common to happen, but it can happen that males and females have an opposite response. And then finally, I would, you know, this is a very interesting, these class 4s, and we have an example later on. This is very interesting because you actually get the same phenotype. And then when you dig a little bit below the surface, you see that actually something completely different is going on under the water. So on the surface, it looks all the same, but actually, you know, when you look under the hood, it's very different. Okay, so what does the data that we get actually look like? And I hope this will be clear just because I'm trying to use very simple graphs to just show you the magnitude of effects. But, we had a paper on a sex difference in how male and female fruit flies respond to diet. So when you feed them a protein-rich diet, females get much bigger. And in males, they don't really care. They don't change their body size at all. So this is a very specific, sex-specific effect of dietary protein on body size. And, I'm not going to go into the details, but basically we figured out this super detailed mechanism in females, where we show that the reason females get bigger when they're fed a protein-rich diet is because they augment the activity of a very conserved pathway called the insulin signaling pathway. It's a hormone that's present in all metazoans. So, we completely, really to the nth degree, figured out why females were getting bigger in a protein-rich diet. But what I can tell you is that other than saying that males don't do that, we actually don't understand what's happening in males. We have no idea yet why they're completely resistant to the effects of dietary protein on body size. So I just want to leave you, each slide, I'm going to leave you with a little message, because sometimes, with all this pressure to study sex differences and to include both sexes, and I really think everybody should be studying both sexes all the time and analyzing by sex. But I just want to give you permission to only figure out what happens in one sex. People feel pressure to figure out everything of everything, and you can't do that in one paper. So hopefully, if you include both sexes and you only know what's happening in one sex, hopefully, today I can give you permission to report that and publish that, because that's okay. Class 2 is more of a sex-biased response. So for example, when we study fat storage, what we found is that we knocked out a particular gene that restricts fat storage. Females got about 40% fatter than they normally would be. But males gained almost, 300% more fat than they normally would have. So males got incredibly, they gained a huge amount of body fat, and females didn't really care when they lost this gene. A little bit, they care a little bit, but not actually very much. And, when we went through again to the nth degree to characterize this, what we figured out was that actually the reason that males care so much about the loss of this gene is because it's present at very high levels in a number of different tissues in their body. Whereas females, they only really have this gene. Mostly its effects are concentrated in the fat. So when males lose this gene, they're losing its function from a number of different tissues and cell types. And that all of those cell types actually are responsible for influencing body fat. And so, this is a really cool paper, because not only did we discover a new function for this gene in neurons, which no one had previously reported, but we also kind of figured out why the effects were so sex biased. So I just wanted to, encourage everyone to, if you see this kind of response, not just to say, because I see this in a lot of papers, males and females both go up. But you can do some very simple statistics to just figure out if the magnitude of how much they increase in their body fat or some phenotype is the same. Because if it's not, there could be some really cool biology. Like in other words, the transcription factor response or something to that gene is much more powerful in one sex. So that sex cares much more about that gene. Or like I said, different tissues or cell types express that gene in females and not males, or the other way around. Okay. So, when we talk about how obvious are sex differences, I just wanted to make a point. So this is a paper that we just posted on bioRxiv, and this is in mice actually, because this is really where in flies, we can detect these smaller differences. A lot of us have been trained to think about differences as though it should be all or nothing. And that's really not the reality of biology, right? Females don't store fat and males store none. Females just store more fat than males, and it's only about a 10% difference. So we really have to change how we think about looking at data and whether or not there's going to be a sex difference and a sex difference in the response, and how big of an effect we should expect to see. And so before I just kind of quickly look through all of these data here, I just want to make the point that we're separating how we display males and females, because we do detect an interaction between sex and treatment. So we know that males and females are not behaving equivalently when we give them a certain treatment. And so here, I don't want you to focus on the details, but what I want you to focus on is basically the shape of this graph. So in females, when we subject them to this drug, this measure of protein synthesis, it goes down in females. And actually, if you look at males, it goes down too. But what you see is that females rebound, they recover, and males do not actually recover protein synthesis when you feed them this drug. So there's a very interesting response there. But if you just look at the shape of the curve, you'd be like, yeah, it's the same. It kind of looks like they're doing the same thing. But when you look at the statistics, actually, they're really not doing the same thing. But, it's not this all or nothing response. So I think it's really important to recognize the magnitude of what you're seeing. Similarly, when we look at how much glucose-stimulated insulin secretion we see in male and female islets that are subjected to a drug treatment, you could just look at the shape of these curves and you'd be like, yeah, that's the same between both sexes. They're kind of doing the same thing. But actually, if you look at the data, what you can see is that over time in treatment, females actually, increase how much insulin they're secreting over time compared to baseline. And then they actually recover after the drug treatment. They return to baseline. Whereas males, they actually don't significantly increase insulin secretion over time. And actually over time in the drug, they actually drop how much insulin they're secreting. So again, even though overall the shape of the curves kind of looks pretty similar and you could go, yeah, it's kind of the same, I really want to make the point that it's really not the same. And even small differences matter. And the reason we know this is because when we know, one sex is going to develop, glucose intolerance, and they're going to develop diabetic-like phenotypes in females and males. So these kind of quote unquote small differences really do matter. And so I'll say something a little bit different from earlier, and maybe we can have a good discussion. And I just want to make the point that, I really do think it's important to study both sexes all the time and to study them separately all the time. And the reason for that is because we recently published a paper on this class 4 difference. And this kind of sat on the side as a project in the lab for a while. And it was because when we gave the males and females a different diet, we saw that the phenotypic response was the same. They both changed their body size in the same direction, same magnitude, everything. But then when we started to genetically pick it apart, we realized that males and females have completely different genetics underlying this decrease in body size. That's what we studied. And so, for example, if you look at this gene ontology analysis, gene set enrichment, overrepresentation analysis that we did on RNA-seq data, what you can see is you see male and females represented here. You see green and purple dots all the time, but there's a lot of categories where you see just a green dot or just a purple dot, or where the dots actually diverge quite a bit. And so that can give you clues that there's something different going on between the sexes. So I'd really emphasize that, you shouldn't be afraid to report a difference. It doesn't make sense that, males and females would accomplish the same phenotype in 2 different ways. But the fact is that sometimes that is what they do. So I think, it's really important that, we all consider incorporating both sexes all the time, and definitely showing your analysis from males and females this differently. So everything people can do right away is separating both sexes for collecting the samples and analysis, analyze the response in females, analyze the response in males. That's how you're going to see if they're doing something different. Use statistical tests to see if there's an interaction between sex and genotype or sex and treatment. And also, please clearly state which sex was used in the methods and the legends. And that will just make it easier for people to know what you did and to reproduce it. And that's all from me.
Wow, thank you. My goodness, I hope that people can give us like a little animated clap, because I think I know our speakers did a fantastic job. And I think we could also talk about this all day, actually. I have so many things that I wrote down in addition to, I'm sure, lots of questions from the audience. So I'll start with something that was posed from the audience and is one of my favorite questions. And then Jennifer will chime in with another question from the audience. And then please, if you have any other questions, put it in the chat or raise your hand, and we'll try to get to as many questions as we can in 16 minutes, which I know won't be very many, which is always the challenge with these types of formats. So one of my favorite questions is, does use, and maybe more towards Ariane, but it can be for anyone, does using sex in the analysis, and I did forget to mention at the beginning that we were focusing more on the sex side of things as opposed to gender, but we're also happy to answer some questions about that. Does using sex in the analysis always reduce power, Ariane? You kind of addressed this, but it's one of my favorites.
I did address it. And no, it doesn't always reduce power. And the answer is it depends, right? So that's like, I don't know. I feel like I could just have like a flashcard. People come to my office and like, what should I do? I'll be like, it depends on like, what's your what's your specific question? What do you think might happen? So in the case where you have completely opposite effects between the sexes, then no, including both sexes won't reduce your power as long as you analyze the sexes separately, right? It'll completely wipe out your power if you ignore sex, include both sexes, they have opposite effects. You don't analyze you don't include sex as a variable in your analysis, it'll actually completely wipe out any difference you might have seen between the groups. So in fact, essentially make your power zero. But as long as you're aware that this could be a possibility, it could actually increase your power. So if the differences between the sexes are large enough, you might actually end up with a greater power than you would have had if you had only looked at one sex. So it really depends on the specific question and what's happening in your study. But yes, if the differences are more quantitative or subtle, like Elizabeth was just telling us about, then
in the last example, she had some really powerful.
She had some very big differences, yeah. But if they are smaller, then yes, it will reduce your power. So I think that's really straightforward.
I think so too. And then Jennifer has the next question, which I think kind of beautifully leads into what your answer just said.
It does. So one of the questions that we got from the audience was about graphically displaying your data. So are there ways to graphically display your data that allow you to consider sex and gender in the way your findings are reported? So if anybody on our panel could illustrate perhaps what you've done in your labs or in your publications that highlight sex and gender, mainly sex, in your findings.
I think Liz, your last one was beautiful, right? Your last.
Okay, the first thing I tell everybody, it's like something so few people recognize and know. If you use any, and so, the statisticians here might cringe and, whatever, but lots of people are using like prepackaged programs. And I hate to tell everybody, but they will automatically set your axes and it will look like there's nothing going on. And then suddenly you realize the female axis is like 1 to 100 and the male axis is like 1 to 2. And you're just like, oh, that's so bad. So honestly, like just get your axes the same. Don't hash them. Don't, to make it look like, get the axes the same. And if you look at papers, check the axes. Because honestly, I cannot tell you how many people are like, they'll show me their data and they'll be like, yeah, so like, here's some of our data. It doesn't look like there's much going on. And I'm like, your axis is completely different. And they're like, oh my gosh, it is. Because in our brains, we just don't, we're trained to look at the graph we get. So I would that's step one, definitely check the axis. And then in terms of graphically representing, I really am becoming a big in favor. I'm not going to try not to monopolize, but I'm very much in favor of showing the treatment effects or whatever effects on females and then showing how the same thing happens in males. Sometimes, we do that with different genotypes often. We use a genotype and we're like, oh, look, I've made a knockout in this. And oh, look, this is how the knockout behaves. And I feel like that does full justice to each sex is when you properly consider how each sex is responding over time to treatment, to genotype, to whatever. Sometimes when you have too much information on one graph, you really lose what one sex or the other sex is doing.
Yeah, and I would also just encourage one of the first things I learned in my stats class was just to graph it. Don't just look at your output, but graph it and make sure you got different colored dots or whatever for males versus females, just to see, do you see that change in the curve or axes or magnitude, whatever the case may be.
Yeah, and I mean, this may sound bad, but that one paper that we published where males did nothing. We have no idea what they're doing. We don't know why they don't respond. So, were the males in the supplemental? Yes, they were. They were in the first part of the paper, but then in every other thing, where literally everything we did, we just, put the females in the main display to display all the data. So if it becomes a matter of having to share a room and, I mean, I'm not impractical, right? You only have so many panels.
I'll ask the next question, which is again maybe more for Ariane and Annalise, but is there a benefit to analyzing sex or gender in data with small sample sizes? And what about unequal sample sizes? So if you had a really small sample, say, I don't know, humans, an N of 10 in each group, or mice 4.
So the question is there a benefit to analyze by both sexes?
Mhm.
Yes, always. I think that's been made clear here, right? Because even if you don't have the power necessarily to detect that difference as being significant, it's important to get an estimate of what that difference might be, as well as you can given the sample size you have.
Yeah, and it feeds back into that issue of maybe your study is not powered to detect those sex differences, and that's something you can describe when you're describing the results of your study. We didn't find sex differences, but we weren't powered to find them.
And I'll say we have a paper, Murphy et al. Is the first author last name, and it was in humans, and it was with mild cognitive impairment versus normal aging. And I think we only had a sample size of 15 in each group, and we saw a sex difference. So even though it was small, we still saw something, and we just called it an exploratory study because it obviously didn't have great.
Well, that's the thing, right? If it's not necessarily powered to detect those sex differences, at least getting an estimate of them out in the literature will help future folks or even yourself the next time you go to design the next project, right? So it absolutely is always useful to analyze by sex.
Yeah. One of the things that didn't come up earlier, but I think is an interesting thing to consider is that identifying and categorizing or quantifying sex differences is sometimes an iterative process. So especially when you don't know if there are sex differences, you may not really power your study to fully explore those sex differences, but then you detect that interaction in your first study, then you have a good indication that that's something that you should explore further. And then, maybe you need to do sex-specific studies. Maybe you need to do studies where you're combining males and females or comparing males and females directly. But whatever that subsequent path is, that first indication is really important to include in the literature.
That's critical. Thank you, Annalise, because I think we so often feel like we have to be perfect, right? Like we have to have the perfect design, the perfect sample size. We have to be able to do everything. But these initial studies where we're just like, we don't know what's going to happen, but let's make sure we include both sexes so that we can at least get an idea of what that difference might be is absolutely crucial.
And then if I could just chime in, I would just say to tell people, just to tell, like the knowledge, I can't tell you how many times people have told me, yeah, the knockout does nothing in females. So we just publish the males. Even if you just report just report that one piece of data, females doesn't do anything. Somebody else can run with that. So it's not bad. It shouldn't be bad. And you should feel free to push back on a reviewer that tells you that your mechanism is wrong if it doesn't work in both sexes. That's complete nonsense. This is for transparency and for like scientific discovery. You should report that negative.
I love that concept of just report, just share what you're finding. And I think that transparency, that's one of the pieces I'd take away from this workshop. Annalise, I do want to jump back to the hormonal phase considerations, considering females, and we'll get to the question in the chat in just a moment. I'm wondering if you can share your tidbits from your presentation about that.
Sure. Do you mean the slides or just the summary?
Summary, I think, would be great for folks to know.
What's your yeah. So essentially, what I would have to say to this is that there are definitely traits that vary with cycle phase, but people became concerned initially about, I'm going to have to include females in my research. Part of the original resistance to using females in research is this myth that females are more variable because of ovarian cycles. And that has been roundly disproven in so many different contexts at this point. The first paper, Prendergast et al. 2014, looks at it in mice. It's been looked at in rats. It's been looked at in humans and gene expression data sets, and in so many different contexts across a wide variety of traits. Females who are unstaged relative to their estrus or menstrual cycle are simply not more variable than males. And it may be the case, and there are several interesting studies sort of looking into this. Where does male variability come from? Is it dominance hierarchy? Is it group housing? Actually, all of these things so far influence variability in both sexes. So regardless, the point is that females are not more variable than males when you ignore staging. Does that mean you should ignore staging? Well, it depends. We all need this flashcard. It depends. Are you interested in how the estrus or menstrual cycle influences your trait? Obviously, you're not going to ignore the stage of the cycle in that case. And we did a study at one point quantifying how much, if you overectomize females, does it reduce their variability? It doesn't. So even taking away that cycle phase doesn't make females more consistent. So for the overall picture of does it influence variability, do you have to be concerned about it? I would say roundly no. We did one positive control to say, hey, does it increase variability? Yes, it increases variability in reproductive behaviors. But outside of this very narrow realm, the daily variability is actually more. And that's the one other thing I'll add, is that you can even look at a trait that is known to vary with the stage of the estrus cycle. For example, sucrose preference varies across the estrus cycle. If you simply take animals across 2 cycles and test them over 8 consecutive days for their sucrose preference, and you know their cycle date, and then you realign the data based on cycle date, you get no improvement in detection of sucrose preference, even though there's this subtle signal with sucrose preference varying by cycle. Why is that? It must be that the daily variation from other sources, who knows what, right? It was a Tuesday. People walked by the hall loudly on Wednesday. Whatever those other sources of variability are, they swamp these tiny sources of variability from the estrus cycle. So is the estrus cycle variability there? Yes, if that's what you're interested in, you need to study it. If it's not the focus of your study, you don't have to worry about it increasing variability in your sample.
The only caveat I'd put with that is, which you touched upon already, which is if there's already a known finding out there, like I'll think of the CA1 spines changing with estrus cycle, and that was William McEwen's work from based in the early 90s, then you'd want to look it up. So I know when I was a reviewer for an NIH grant, they said something about like, we're not going to look at estrus cycle because food preference doesn't change with hormone levels. And I'm like, I think that you might be off on that one. So just if there is literature out there, important to know about it. But I agree that the variability is completely the same between males and females, and males will have a diurnal fluctuation too. Sorry, I think I went off the course there. Jennifer, you're next, I think.
Yeah, there's a question just in the chat here that I'm wondering if any of our speakers want to speak to about understanding the effects of sex and gender and hormone replacement therapy within trans individuals and their response to various drugs and treatments. So thinking about this as a unique population, that maybe we tap into kind of our discussion on sample sizes and making sure that we're getting a good sample size for this group. But wondering if anybody wants to speak to that.
How would I rephrase it? Which may or may not help. My brain is not obviously working as well as it should right this second. But so would you expect in a trans individual for a treatment to be the same as in a cis individual?
It depends.
Yes, I knew that's what you were going to say.
Maybe. I mean, I'm not, yeah, it could be different, right? And we don't necessarily know what that would be. And that is one of the worst things about not having included enough trans people in these studies to date, right? Is that we don't actually know. And so please get started on that. Whoever it was who asked that question. The best way to figure that out is to start including trans people in studies, looking at drugs and seeing if their responses are the same as cis individuals.
And I'm sure it depends on whether or not I'm a little biased because I always think that sex and hormones play a role in virtually everything, but that it'll depend on the gender affirming therapy that they're taking at the time as well. So
absolutely, and how much they're taking, like what's their dosage, how long have they been on therapy, right? So longer somebody's been on therapy, the difference obviously 100%. Will be in how their tissues react. Mm-hmm. And gender is going to be a whole other can of worms, and starting to talk about the social impacts of gender on people's health. So I don't think that's what we're talking about today, but that is a whole other layer that you would have to add to this once you start talking about people and gender.
Mm-hmm. Yeah. And go ahead, Jennifer.
Sorry, just on that, I think one of the key things that we're hearing also is just report your data, share if there are trans individuals in your studies. Why not look at that question to see if there are differences between trans, cis, between what your population looks like. So that would be one takeaway that I would have. Lisa?
And report, and I guess like you said, report, display the data so in a way that people can use that data to further investigate it down the road. So with that, we're actually at time, even though we have many more things to say. I'm sure there are many other questions. As I said, I wrote down like 20 on my page. So I don't know how, we'll just have to have more of these workshops so that with more time for questions. So thank you so much for joining, and please join me in thanking our amazing speakers just at the start of the conversation. And I think the answer is, it depends. And don't be afraid, report.